Hello students, this is Ritik Sir, your biology SME. So in this video, I'm just trying to summarize the entire chapter, that is sexual reproduction in flowering plants. So it's obviously a very huge chapter, but I'm just trying to summarize it, okay, in detailed or very specifically. So it's a kind of revision video, so where you can just watch in a one shot of revision, okay. So yeah, coming to the sexual reproduction in flowering plants, what are the major aspects is we know when in the plant kingdom we have been studying like other things, right? On the evolution aspects of the plants. So the last position you'll actually see the evolution of flowering plants, where in a proper flower you will be finding both male as well as female reproductive system. So flower, you can actually see all like both kind of reproduction. That is, asexual reproduction can be seen in some budding is also being observed, and basically sexual reproduction is also observed. So whatever the flower plant which is able to produce the flowers, right? So they actually will be seeing the sexual reproduction in those plants. That is, most of the flowering plants they reproduce their offspring through the sexual reproduction. So that is one aspect. So what about the male and female sex gametes? Here, we in the flower, okay, we will be having both the male gametes as well as what female gametes. So when I'm talking about the male reproductive and female reproductive system, we actually have this called androecium, and the another one is sorry gynoecium. Okay. So one is androecium and another one is gynoecium, right? So what do you mean by the androecium? So this whole stamen part, right? So it has been actually represented by androecium, whereas this carpel, right? So that is which normally represent male reproductive system, and gynoecium represents this female reproductive system. This part has actually been represented by what? Female reproductive system entirely from here to here. Whatever you're seeing is actually being represented by female reproductive system. So first we'll try to understand about the male reproductive system. So in male reproductive system, what we do have one is stigma, just a minute, style. So if you see a section of this plant, right? So what are the things that you are going to see? And I'm talking about the male reproductive system, that is uh, androecium. So here you'll actually see in androecium, you'll see the stamen part basically, right? So this is actually called the entire part, okay, is actually called as stamen part. This entire part, what I'm talking, is called as stamen part. So in the stamen, you'll actually see the filament. So can you see this filament? So one thing you will see is the filament you will see, and the upper part, this thing, you'll actually call it as anther. Right. So what are the things you are going to see? Two parts you are going to see. One is filament and anther, right? So yeah. So basically filament is which is long, slender kind of thing. And that terminally generally bilobed structure. This is called as anther. So that is to the filament, anther is attached. So this is one point. So if you observe them carefully, you'll also see that this filament and anther, that is the entire stamen part, is not equal. That means you won't see the equal length or basically equal length in the microscope. So there will be a vary of its length. So what is the speciality now? What is the speciality? See, if I'm talking about the anther, see, we have a thing called here, that is in the. So we are actually talking about angiosperms, right? The most evolved plant. So in the angiosperms, the anther is actually bilobed, which each lobule, that is, this is the bilobe, where each lobule is having two theca. Okay. So they are actually called as thecus. So as you can see here, so each lobule will be having two theca, which we normally call it as dithecus, lean of dithecus. Okay. And another important thing is like when you take a transfer section of this anther, so what you are going to see this thing where in the pollen sac, pollen grains generation happens. Okay. So the anther, as we can see, if you take a transfer section of this anther, so what you're absorbing here, you're actually absorbing four-sided structure. So this four-sided structure, it consists of what? Microsporangia. Okay. So this four-sided structure consists of what? Microsporangia, which is located in the corner. Where they are being located, guys? They are being located in the corner. Okay. uh, like two lobes, two in each lobe. That is, if you're talking this is one lobe, you will be seeing two microsporangia. In this lobe, you'll be seeing again two microsporangia. Okay, this is one thing. So in this microsporangia, what you're going to see, the microsporangia, further, it is going to develop to become what it becomes, to become the pollen sac. Okay, this is very important. These microsporangia will further develop to become what? Pollen sac. Okay, and in the pollen sac, what you are going to observe, so the pollen sac are packed with the pollen grain. Okay, so this is about the basic about the anther. So now another very important thing is you will come to know the structure of the microsporangium. That is, I'm talking about microsporangia, right? So how exactly the structure looks like? Yes. When talking to the structure of this microsporangium, say this is microsporangium. See, when you take a transfer section of the microsporangium, right, it nearly appears to be a typical circular in outline. This is one point. Then you will be seeing four kinds of walls are present, right? So what are those walls? See, if you take a transfer section of any one microsporangium, you will see somewhat circular thing. And you have four walls. One is epidermis. This is one. The next one is endothecum. This is the second one. And the another one is the middle layer. Okay. So first one, epidermis is your first one. Then the second one is endothecum. The third one is middle layer. The next one is tapetum. Okay. So these are the four layers that you are going to observe in the microsporangium. So out of this, the outer three wall layer performs that is outer three wall layer performs the function like protection and helps in the disintegracy of anther to release the pollen. That is, one is protection and anther to release the pollen grains. Okay. So this is major function of these three layers, that is epidermis, endothecum, and the middle layer. So now what happens? This tapetum that I'm talking, which is actually your fourth layer, right? So what is its function? So nourishment. That means, as you can see, there are microspore mother cells which are going to produce the pollen grains, right? So, for the development of pollen grains, it requires some of the nutrients, right? So, that nourishment is actually provided by tapetum. So, nourishment is the major thing. Okay. Then cells of these tapetum, as you can see, they are the cells, right? Cells of these tapetum. Okay. So they will be possessed dense cytoplasm. Cells possess dense cytoplasm. And another one is generally it will be having more than one nuclei. Having more than one nuclei. So these are the major things. So when the anther is younger, so what happened? They are actually being arranged. When the anther is younger, okay, uh, a group of compactly arranged homogeneous cells you will be observing. So these group of compactly arranged homogeneous cell is actually called as sporogenous tissues. Okay. So can you see here? Here you will be observing compactly arranged sporogenous tissues. Okay. This is one thing. So they normally occur where, as you can see its position, it normally occurs in the middle, that is center of the microsporangium. It normally occurs in the central of what? Microsporangium. So these, that is the micro, and coming to here, that is microspore mother cells you will be observing, right? So they undergoes, as you can see here, the sporangium tissues, okay, as a result, which has been in middle, right? So this sporangium tissues, they undergoes what? Meiotic division. Okay, they undergo meiotic division, and as a result you will actually see to form microspore tetrads. Okay, so as a result to form what? Microspore tetrads. This is again one thing. So now what happens? As a result, this each cell is able to of this that is the sporangious tissues, where each cell in those tissues is capable of giving rise to what? A microspore tetrads. Okay. And these microspore tetrads, okay, each will be having the micros. So this is actually the microscope tetrads where you will be seeing this microspore mother cells. And these microspore mother cells, right? So they form, that is microscope mother cells or pollen mother cells. So these microscope mother cells forms pollen mother cells, and these pollen mother cells again through the meiosis process. Okay. They go through meiosis process, which is actually called as microsporogenesis process. So this is one important thing. Microsporogenesis. What is microsporogenesis here? The process of formation of the microspores from the pollen mother cells. The process of formation of the microspores from the pollen mother cells through what? Through the meiosis division. Okay, through the meiosis is actually called as microsporogenesis. Clear? So these microspores, as they are formed, they are again arranged in cluster of four. That is, They will be arranged what in a cluster of 4 cells, which normally I call them as microspore tetrad. Okay, microspore tetrad. So now what happens as a result? The anther gets matured. So as the anther gets matured and dehydrates, okay, so these microspore which gets disassociated from each other. So that is the microspore which was together, right? Now they gets disassociate with each other and develops into the pollen grains. Getting. So now what happens? It is going to develops into the pollen grains. So now you will find the pollen grains. So now another very important. So yeah. So here you can actually see a microscopic structure of Microsporangium. Okay, where they are been associated together. So once they get matured, they get dehydrated and they move from each other. And as a result, what happens? So they are going to form an individual pollen grains. So variously, what happens is from plant to plant, from flower to flower, the structure of the my this pollen grains differ. Okay, that is again one structure and color design of the pollen grain gets different. The basic thing that we need to know is one thing we need to know is the all over structure of a pollen grain. So the outer hard covering, you're getting the outer hard covering or outer layer is actually called as exon. Exon is actually made up of sporopollenin. So what is the sporopollenin? It is one thing is it has some more resistant organic material. No. So that means it helps in withstanding of any temperature, pH, acidic condition, whatever it is. Okay. So that's why it won't allow the pollen grains to get destroyed easily. This is one thing. So it can withstand the high temperature, strong acid and pH and alkaline. So no enzymes are yet found which could degrade the sporopollenin, right? But in the pollen grains, that is in the exon region, in some places you will actually find a pores, a prominent appearance you will find. So they are actually called as germ pores. Germ pores. So where in these germospores, what happens? These sporopollenin is absent. So now, again, there is something very unique in our nature. This sporopollenin will not allow, it has a resistance towards high temperature. It can withstand high temperature. Then again, it has some resistance towards the acid, alkaline, pH, something like that. So this is one condition. That's for it. It won't allow the pollen grains to get damaged. But again, in some part, you will actually see the germ pore where the sporopollenin is not present. So that means that is the part from where the pollen grains can be destroyed. This is one thing. Okay. So this thing you need to remember. So because in the pollen grains of outermost part, you are actually seeing the sporopollenin, the pollen grains can be preserved for a long duration because they are unaffected for a longer period of time. That's why the pollen grains can be preserved. This is another one important thing. Then we have inner wall here. So inner wall, which is actually made up of intin. Okay. So intin and continuous layer, inner wall of the pollen grain is actually called as the intin. So in intin, what you are going to see? You are going to see two types of cell. One is vegetative cell and another one is generative cell. The vegetative cell, as you can see, it is somewhat bigger when compared to the generative cell. The vegetative cell is somewhat bigger when compared to the generative cell. So it has an a nucleus here. Okay, generative cell. So this generative cell is small and floats. This generative cell normally it floats in the vegetative cell. Okay, and you can see here the spindle fibers with the dense cytoplasm in the nucleus also. So you will see a two cell stage here. So that is this is vegetative cell and this is generative cell. So now what happens? The pollen grains, okay. So pollen grains are shed at two celled stages. That is, pollen grains, they come out from the anther in two celled stages. That is, one, it will be having the vegetative cell and another one, it will be having the generative cell. Okay. And in the remaining species, what happened? The generative cell, it normally undergoes the mitotic division to give rise to two male gametes before the pollen grains are shed. Okay. So that means in pollen grains, obviously you'll be seeing now three celled stage you are going to see. But somewhere these pollen grains are also causes some inflammatory disorders or several allergic disorders, allergic reactions in some of the animals as well as human beings, such as chronic respiratory disorders, bronchial disorders, asthma and all. But again, there are some advantages. Okay. It's like since I have already mentioned that the pollen grains is very rich in nutrients, so they are being used as the pollen capsule. Even pollen syrups are being used. Okay. So these are some of the economic importance of the pollen grains. After the completion of the male reproductive system, so we will move to the female reproductive system. So in female reproductive system, it consists of what? So pistil, megasporangium, which is nothing but the ovule and the embryonic sac. So female reproductive system is nothing but gynoecium, right? Uh, in the gynoecium. So this is actually what they are. When you take a dissection of a flower, you'll actually find the piston here. Okay. So in the pistil, you'll actually see these things that are stigma, style, then you have ovary and thalamus. So thalamus is the last part where ovary is attached anyhow. So this is stigma. So once the pollen grains dispersed from the anther, so that pollen grains will come and it is going to sit on the stigma. Okay. And I'll explain you in detail what all the things happens. So from stigma, what happens? The pollen grain, as you can see, so you will see the development of the pollen tube also. So through the pollen tube, okay. So by the pollen tube, through the style, so the gamete is going to release its what do you say that the nucleus, and that is going to reach to the uh, ovule, which is present in the ovary, and fertilization going to take place. Cool. So this is the major thing. So as I told in the pistil, you have the ovary. Within the ovary, you will actually see this ovule, which is nothing but megasporangium. So the megasporangium, when I'm talking about the megasporangium, the ovule is very small structure which is attached to the placenta region, which is attached to the placenta by the mean of stalk, okay, which is actually called as funicle. So now I'm talking about what? This thing called ovary. Oh, sorry, ovule, which is also called as Microsporangium. This part. So now the thing is this ovule that you're seeing, that is the embryo sac, the ovule. So basic this ovule, entire ovule is actually very small and it is attached to the funicle. Okay. So this is one thing. Then you will also see this funicle is being fused with the hilum region. So this thing, funicle is fine. So we know that the body is again fused with hilum region. Thus, hilum is one thing which normally represents the junction. It normally represents the junction between what? Ovule as well as funicle. Okay. And each ovule has one or two protective layer. Okay. Or one or two protective envelope. So those envelope are actually called as integuments. So here we have inner integuments and outer integuments. So that's what. So each ovule will be having one or two integuments, that protective integuments. So integuments, okay, that is, it encircles this nucleus. As you can see, this is the nucleus. So this integuments, they normally encircles or covers this nucleus except the tip of the region. Can you see the tip of the region? So here it doesn't covers in the tip of the region. So this is one thing. So there in the tip of the region, what you're actually like observing, you're observing a microscopic or a very small opening, right? So you're actually observing a small opening. So that opening is actually called as micropyle region or micropyle. Okay. So that opening is called as micropyle. So this micropyle will always be opposite to this chalazal pole. So if this is the chalazal pole, to this opposite only, this micropyle region will be present. And below the micropyle region, you will actually see the micropylar pole. This is a basic thing. So the integuments actually encloses what? The nucleus. And the nucleus, it will be having abundant reserve food material. Food material. Can I say this? So there, this thing you will be seeing. So located in the nucleus is the embryonic sac. So in the nucleus, what it is located? Embryonic sac is located in the nucleus. So this embryonic sac is also called as female gametophyte. Okay, so it is called female gametophyte. Ovule generally have a single embryo basically. Every ovule will be having single embryo. So which normally going to form what? Megaspore. So this is all about the ovule. So now let me see how this embryonic sac development takes place. Curious? Are you curious to know how embryonic developmental takes place? So let's see. So the diagram where they have mentioned, see, I'll just try to explain the process. So say if this is the embryonic sac, so how the development is going to happen? The process of formation of the megaspore from the megaspore mother cell. Okay. So this is the megaspore mother cell. So what is the thing? The process of formation of the megaspore. Okay. Megaspore from what? Megaspore mother cell is actually called as what? Megasporogenesis. Getting. So it is actually called as megasporogenesis. Clear? So right. So ovial, what happens? It's generally get this thing differentiated into a single megaspore mother cell. This is one thing in the micropyle region. So if this is the micropyle region, so what happens? The ovial gets differentiated. So of the nucleus. So what happens? So once the differentiation happens, so you will see one thing is you are going to see the megaspore dyad. Okay. So that is the large cell contains now the dyad region. So two things you'll be seeing, and you are also going to see the nucleus around it. So one way you are going to see the nucleus. And this is the micropyle end region. So the cell will be containing what? It will be containing a dense cytoplasm, and the prominent nucleus will also be absorbed now. Okay, the prominent nucleus will also be absorbed. Again, this megaspore mother cells, so again it is going to divide meiotically. Okay, meiotical revision. So as a result, what happens? You will see the megaspore tetrad, and finally you are going to see the stages. So female gametophyte development, what the thing is, the majority of flowering plants that we are talking about, so they're functional while the other, so in this thing that is tetrad, as I told you, so they'll be producing the tetrad spores, right? So in which tetrad megaspores, right? So in which majority of the three will be inactive, getting. So other three will be degenerated, where one will only be the functional here. Okay. So only one will be the functional megaspore. So which later on going to develop into the female gametophyte or embryo sac, something like that. So there what happens, we will be seeing the megaspore development from that way. See, as now you can see that you'll take this one cell now. So what happens? The nucleus, which is actually the functional megaspore. So you have been seeing 1, 2, 3, 4 megaspore, right? So we called it as megaspore tetrad. As I told you that in these four, only one will be the functional megaspore tetrad, where other three remaining will get degenerated. So now the functional one, as you have already seen, there will be only one nucleus, functional nucleus, right? So they undergo again what? Meiotic or mitotic. What division you'll see? Here you are going to see the mitotic division. Okay. So here you will see the mitotic division. That means it undergo mitotically to form how many nuclei now? Two nucleated embryo sac. So this embryo sac has now two nucleus. And these two nucleated embryo sac, again it undergoes the meiotic division to form what? Four nucleated embryo sac. And this 4 to 8 nucleated. Again, year 4 and year 4. 8 nucleated stage of the embryo sac. So total you will be having how much now? 8. Okay. So now in an instant, these mitotic division are strict only with respect to the nucleus. Okay. So there will no be cell division and all. Only nucleus will get divided. And once the nucleus get divided, right, immediately cell formation will not happen. Gradually the cell formation, cell wall formation will happen. Okay. So the female gametos like embryo development, you'll start seeing now. So as you observe here, so what happens? Out of eight, 6 out of 8, the 6 nuclei are, so that is say like 1, 2, 3, 4, 5, and 6. 6 nucleus are being surrounded by the cell wall. Can you observe here? So 1, 2, 3, 4, 5, 6. So 6 are being surrounded by what? Cell wall. And they are being organized into a cell. They are being properly organized into a cell. So whereas remaining two nucleus which you have been seeing here, right? So they are termed as what? Polar nuclei. They are being termed as polar nuclei, and they are situated where? They are being situated in the central of the cell. Okay, they are being situated in the central of the cell. This is another point. So now what happens? So you'll be seeing the equal distribution here. So the cell, there, the three cells here that you are seeing, right? They will be grouped together here and here. So these one, two, three cells will be grouped together at where? This microphile end, and constituents what? The egg apparatus. Okay. So the egg apparator in turn, what happens? The egg apparator in turns, it constituents this thing, that is two synergids and one egg. Okay, two synergid and one egg cell. The synergids here will be having a specialized cellular thickness, which is actually, you will find in the tip of this microphile end, which is called as filiform apparatus. So, which is going to play a very important and major role in guiding the pollen tube into the synergids. Okay. So, three cells, as you can see here, this is one point. Next, here you are seeing another three cells, right? So, three cells were in the chalazal end region. So, they are called as antipodals. Okay. So, in the middle, you will actually see a large central cell which is present in the middle, as mentioned. So, where you will be observing two polar nuclei will be observed. So, thus, typically what happens, the angiosperm embryo sac at maturity, though it will be having though it will be having eight nuclei, that is one, two, see, like you, even though you have eight nuclei, so this is one, two, three, four, five, six, seven, eight. Even though you are having eight, what do you say that? Nucleus, but how many cells you can observe? Only seven cells will be observed. Seven celled, eight nuclei or nucleated. So this is all about what now? The female reproductive system. Topic that we have is on pollination. Like now we get to know about the male and female aspects of the reproductive system in the flowering plants. So how pollination happen? So that is the question mark, right? So how exactly the pollination? What are the means of pollination? And later on we will try to understand the mechanism of the pollination. The first thing about pollination is it can be achieved both, that is, it can be achieved by both mechanically and naturally. So there are some objectives of having in a pollination. So one thing is transfer of pollen grains from the anther to the stigma, okay, where the anther is going to shed the pollen grains, and that has been transferring into the anther to the stigma, right? Stigma where on the pistil part of the stigma. So this is actually termed as pollination. So, so this is what we normally term it as pollination. This is one point. So there are different kinds of pollination achieved. So one such is autogamy. Okay. Autogamy is one such. Then we have is titanogamy is one such. And the last one kind of pollination that we see is xenogamy pollination. So what is this autogamy pollination now? See, it's a type of a pollination where it is been achieved within the same plant, same flower. That is, transfer of the pollen grains from the anther to the stigma of the same flower of the same plant. Okay, of the same flower of the same plant is actually called as autogamy. So in these types of plant pollination, what happens? So the stigma, anther and stigma are completely, they have been placed close to each other, and flower might be closed during the pollination. So that's why there might be a transfer of the pollen grains from anther to the stigma within the flower of the same plant or same species. So this thing could be happen. So this is normally we see on what? Autogamous. So here you will actually see a self-pollination. So self-pollination can be occurred. So there are some common examples such as viola and many other examples of flowers are also being there. So they flowers will be producing two types of flowers. So what are they? One is casmogamous flower and the another one is clustogamous. Clear? So what is this casmogamous flower? See here what happens, they're similar. That means here the flowers which are similar to the flower of the other species. They expose anther and stigma. And when coming to the chias clustogamous flower, what happens? So here you will be seeing the flowers which do not open itself. So therefore the pollination happens within the flower. So they are called as clustogamous flower. And cross pollination is almost prohibited in the clustogamous. Okay. Then we have gytenogamy. So here the transfer of the pollen grains from anther to the stigma. Okay. But in the different flower, but of the same plant. Here what happens, the cross pollination you can somewhat see, but somewhat you won't see. Because since the pollination is again happening within the same flower, right? So involves the here pollinatory agents are also involved here. So but it is somewhat similar to the autogamy since the pollination is happening within the same species. The next one is xenogamy flower. That is xenogamy pollination where the pollination happens between the different flower of different plant. So that is called as xenogamy. Here, cross-pollination, almost self-pollination will never happen. Cross-pollination and, pollinatory agents, they play a very important role. And here you can actually see or you can actually bring up the genetic differences through the type of pollen grains to the stigma in that way. That means you can actually expect something genetic variations can be observed in the coming generation plant. So this is all about the pollination. So there are certain agents of pollination like say wind, water, as well as some of the biological agents are also there. Say if I'm talking about the wind, you are familiar about the corn cob. So what are the things that if I'm talking about the wind pollination, so what all the things that it should contains? So first point it should have is feathery nature. That is the for the pollination, right? So it should have the air. What do you say that pollens should be easily dispersed from the anther. This is one thing. And the pollen grain should have the feathery nature so that they can be easily carried out through the wind current. This is another thing. And they should be lightweight. So this is very important points when coming to the wind pollination. So we have another, that is the biological agent. So that is insects, animals, they play a very major role in the pollination also. So what are again how the pollinatory flower should be? So the pollen grains should be sticky, right? And it should be more attractive. That means the flower should produce inflorescence. It should have nectar so that the insects they could come and they could get the nectar. As a result, the pollen grains are being carried by the insect from one plant flower to the another flower of the plant, something like that. And another thing we have is the water pollination. So in water pollination, what happens? We actually take some examples of Valsinaria. Okay. So here what happens? The female flower, the female plant, it reaches out the surface of the water as you can see here. And later on, how through the help of these large stalks and all, and the male flower, it is going to produce the pollen grains and released on the surface of the water. As a result, what happened? The water current, it is going to carry the pollen grains, and some of them eventually they are going to reach the female stigma of the female plant, and as a result pollination happens. This is one point. So there is other example called plants such as sea grasses. So where you will see the female, it remains submerged in the water itself, and there only what happens, the male plant will also produces a pollen grains which is submerged in the water itself, which is going to produce within the water itself, okay, inside the water. So pollen grains in many species, so they will be, the pollen grain structure will be like long, then ribbon like structure, so that they can be easily carried inside the water. And some of them they will eventually reach to the stigma of the female plant, that is sea grasses, and pollination happens. So this is one way where inside the water pollination happens. This is how the pollination has been achieved. Okay, so water pollination, they don't require these things called nectar, sticky, and all. So basic thing, the pollen grain should be waterproof, and what are the means it should have? It should be long, ribbon-like structure should be there in order to they could like easily carried out through the water current. Something these characters are major or very important, right? The mean of how the pollination occurs. So there is something called outbreeding device concept, where if in order to stop the pollination, unwanted pollination, there are certain way where the plant they have evolved some mechanism in which they can stop the contact of the stigma, that is the pollen grains, okay, so of the same flower. So that is actually called as outbreeding devices. So see what happens is, if inbreeding happens, so as a result inbreeding depression will be occur. That means like say in human beings also, like if we marry to our own cousins or our own sister, something like that, what are the disadvantages of it? you have also seen many genetical disorders can be occurred, infertility problems can be seen in future or in the next generation, right? Same thing with respect to plants. So if there is an interbreeding is happening within the plants, self pollination is happening within the plants, they might lead to what, a kind of inbreeding depression. So where in next coming generation infertility can be explored, or there might lose producing of more whatever the fruits or vegetables that we wanted, so that production will become depleted, something these things will be absorbed, or that they will be okay with, they will be exposed to many disease causing agents. So just to overcome this, the plant have a certain mechanism, we call it as outbreeding device, outbreeding devices. Clear? So in outbreeding devices, we have around four outbreeding devices. Like first thing is, in some species, the pollen which releases and the stigma is it is actually a receptivity. That means it normally receives the thing, right? So they are not synchronized. Okay. Pollen released and stigma receptivity is not synchronized. That means when the pollen is released, the stigma will not be able to receive it. Or when the stigma is ready, the pollen grains will not be released in the same flower or in the plant. So in this way, the self pollination can be avoided. So what happens when a pollen grain is released in a flower, and that flower stigma is not showing the receptivity? As a result, the pollen grains will move to some other flowers or to some other plant flower. So in this way, what happens? The guarantee of outbreeding, the guarantee of crossbreeding takes place. This is one point. So the another thing is the anther and stigma. Anther and stigma are placed, they are being placed on different positions. So they are being placed on the different positions here. So in that way also self-pollination will be controlled. The second one is it prevention of the autogamy. Prevention of gamete. So the third device is what happens? A device to prevent the inbreeding is self-incompatibility. So third point is self incompatibility, right? So in incompatibility, self incompatibility, what happens is the pollen germination or the pollen tube growth in the pistil. Okay, so it has been stopped. That is, it has been prevented now. So another device is unisexual flowers. Like in papaya and all, you will see only in one flower, only the male gamete will be present. And in another flower, only the female gamete will be present. So in this way also, you can avoid the, inbreeding depression. So these are the basic thing. So after this, okay, so now what happens? The pollen grain has been transferred on the stigma now. As you can see, the pollen grain has been transferred on the stigma, right? So the pollen grain has been transferred on the stigma. So when I'm talking about the pollen pistil interaction, so it is the interaction of the po pollen grains which with what? That is stigma. So here you can actually observe the stigma here and the pollen grains. So the interaction of the pollen grains with the stigma is actually called as pollen pistil interaction. So where it begins with the pollination and ends with the fertilization. So at the event, what happens? The pollen grains on the stigma, right? So that entry of this pollen tube into the ovule you are absorbing, that is actually called as synergids, right? Are referred as what? Pollen pistil interaction. Here what happens? The deposition of the pollen grains on the stigma to the entry of the pollen tube are referred to as pollen tube, where in the ovule is actually called as what? Pollen pistil interaction. So pollination does not gradually occurs here. You have to remember that the pollination, this pollination does not guarantee the transfer of right type of pollen. So often wrong types could also land on the stigma. That is, in during the pollination, sometimes wrong pollen grains will also land on the stigma. So what happens? So the question comes as if, so does pollination happens? Say if there is some other species, okay, and that like lily or some jasmine, something. So even they produces the pollen grains. So say does if these pollen grains does it come and sit on the hibiscus stigma, then pollination occurs? No. So what happens? These stigma, right? So there are certain receptors there. So these receptors, they are going to recognize once the pollen grains comes and sits on the stigma, the receptors on the stigma are going to recognize. So there are certain they are being mediated by certain chemical components of the pollen grains. So that helps in the interaction of the pollen pistil. So if it is unable to recognize those chemicals, so as a result, it will not allow the pollination. It will not allow the growth of the pollen tube. So we can now call it as for in so that is called as like they are from other species, okay. So now it once it has been not recognized, so obviously pollination will not happen, okay. It will once they are not recognized, so the pollen grains will be rejected from the stigma, so fertilization will not happen. So once the fertilization happens, as you can see, so they get entered. So after some while, what happens? The pollen tubes development can be observed. So one thing that you can do is you can actually do an experiment. So if you take a pollen grains in the on the glass slide, and if you add some say sucrose solution, and if you keep it for some time, and you close the cover slip, and if you observe it under the microscope, you'll actually see a kind of pollen grain. So if it is say like so if it is this is the pollen grain. Okay, if this is the pollen grain and you have added the sucrose solution and you leave it for some times, so as a result, you will actually see the pollen tube growth also. So another question is if one time, if say two, three pollen grains are produced, will the pollen tube, so all will be having the pollen tube, so will all is going to develop the pollen tubes on the where? The stigma part. So the answer is no. So what happens out of all this pollen grains? Only one pollen tube is going to reach. Only one pollen tube is going to reach to the ovule. Okay, through this micropyle region. say like synergids, it will reach us to the synergids and then to the ovi. So something like that. Okay. So through the micropyle region to the synergids. So later on, what happens? So this pollen grains, it has nucleus, right? So that is going to release one of its nucleus and fertilization takes place. So we have hybridization, that is artificial hybridization. So one of the major way how you can, hybridize, how you can get the desired character through the pollination is, see, artificial hybridization. In artificial hybridization, to make sure that only the desired pollen grains are being used for the pollination, the stigma is being protected from some contaminations, unwanted, desired pollen grains. Okay, so this is achieved by two things. One is emasculation, and the another one is bagging. So, what is this emasculation? See, in emasculation, if the flower has bisexual flower, that means if both male and female are present. So, you are going to removal of anther from the flower with the help of the forceps or something before they discharge the pollen grains, before they discharge this pollen grains. So this is actually called as emasculation. Okay, so emasculated flowers have later on they will be covered by the butter paper, say of the size of some butter paper. Okay, a suitable size or bag of generally which is made up of butter paper, so that will be covered where to the stigma till what time they become they till they become receptive, that means they will be able to receive the pollen grains. Then what happens, the desired pollen grains will be dusted on that particular stigma and the pollination is make sure happen. So this is how artificial hybridization has been done. Okay, so after, once the pollen grains has been, dusted on this stigma, so again it has been rebagged and it is allowed till the fruit development is observed. So if that is not there, there is no need of rebagging and all. Right. So this was all about how the artificial hybridization happens. What might be the next topic? It's all about the double fertilization. That means, so now how the fertilization actually happens. See, what happens here is in double fertilization, right? So in the double fertilization, as the after entry, that is after entering one of the synergens, what happened, the pollen tube, right, it is going to release the two male gametes, because in the pollen grains, as I have already said you, there will be two male gametes. So through the pollen tube, it will be the two male gametes will be released into where the cytoplasm of the synergens, into the cytoplasm, right, into the cytoplasm of what? Synergids. Synergid, right. So one out of these two, one of the male gamete is going to move towards the egg cell and fused with the nuclei of that egg cell. And as a result, where in the synergens, as a result, you will actually see what? you're going to see the formation of the zygote. Okay. So as a result, you will be seeing the formation of the zygote, which is actually diploid, which is diploid cell now. Okay. So what about the other male? So it moves towards the two polar nuclei. It is going to move towards the two polar nuclei, which was present in the, see, one of the uh, nuclei is going to move towards the synergens. So there it is going to fuse with another cell, and as a result, what you will be seeing, you are going to see the zygote formation. This is one thing. So another, I told there was another male gamete, right? So that is going to move towards the center, and it is going to fuse with the center cell. Okay, it is going to fuse with the two polar nuclei which was present, which was located in the center, to form primary endosperm nucleus. So it is nothing but PEN. Okay. So as what I'll be saying is, as this involves the fusion of three haploid nuclei. So that means here you are actually seeing the fusion of how many haploid nuclei here? Three haploid nuclei. Okay. So therefore I call it as triple fusion. Okay, so therefore I'm calling it as what? Triple fusion. So one of the polar nuclei will be, moved to this nucleus, fuses in the synergent region, forming the zygote. This is one point. Second, another nuclei. So where that is going to move to the central, where already you were seeing the two polar nuclei located in the central cell, fuse with them, and it starts producing the triploid primary, uh, that is endosperm. Because in center we already have how many nuclei? Two nuclei. Another one nuclei is from the male gametes. So how many nuclei have been formed? Fused. Three nuclei have been fused. So it is called as triple primary endosperm nucleus. Clear? As this involves the fusion of three haploid nuclei, so it is actually called as what? Triple fusion. Okay. Since how many types of fusion you are seeing? Two types of fusion you are seeing. So one fusion that you have seen in the synergids region, that is the zygote development. Another fusion that you have been seeing in the central part, where the primary endosperm nucleus development or production. So how many? Totally two types of fusion you are seeing, right? So one is syngame. So this is actually called as syngame fusion. And another one is, what do you say that? Triple fusion, right? So as a result, where it is actually taking place, it is taking place in the embryo. Hence, so how many fertilization you are seeing? Two fertilization. Hence, it is called as double fertilization. So, clear with what is exactly mean by double fertilization, right? So here, as a result, what happens? This PEC, right? So and develops into the endosperm, and later this endosperm, while the zygote develops into the embryo. So as you can see here, the zygote part, right, will be developing into the embryo region, and this thing, that is the PEN region, as you have seen, will be developing into the endosperm. Clear? So this is all about the double fertilization. So I told you one thing, that is one is going to develop into the embryo, the zygote part, and the another one, which I told, the primary endosperm cell is going to develop into, so that, and the development into the endosperm. That is what I have said. So what is endosperm then? See, primary endosperm cells, which have that you have seen. So is actually a result of the striploid endosperm tissues. In general, the cell of these tissues, like when I'm talking about the endosperm development, the cell of these tissues will be actually, filled with the reserve food, reserved food materials. Okay. So reserved food materials, and they will be used as a nutrients while the development of the embryo. This is one thing. So what happens? This spen is actually undergoes with the successive endosperm development, as it is actually called as prenuclear endosperm. Okay. So subsequently, you will actually see the formation of the cell wall, and endosperm will start becoming what? Cellular, the wall. Okay. So the number of the free nuclei formed before the cellularization varies. That means number of the nuclei which is present gets varies from one to the other. For example, have you seen the coconut water? Right. So what happens here? Not the free nuclear endosperm, right? So there are thousands of nuclei. The coconut water that you are seeing actually is a nuclear. So it is actually a thousands of nuclei, and they are being actually surrounded by the white kernel, which is the cellular endosperm. This is one way. So not like everything has to be something like that. So during development, that free nuclei which is there, right? So that will be utilized, okay, by the embryo during the development. This is one point. So that's what I'm saying you. And even during the development, the embryo might be consumed completely, or endosperm might be consumed completely by the embryo or not completely. Okay. So it depends during the development. For example, if you take if they may neither be, for example, if neither of them might completely consume this endosperm, such as in some examples I'm just giving you, an example like pea or groundnut or beans and all. So what happens before the seeds get germinated or the seeds get matured, right? So it may persist in the mature stage. Okay. So whereas in so there what happens? So they won't consume completely. But if you see in some like coconut and all, you will be seeing that it may persist. That is, the endosperm may persist in the matured seed. So that means in matured seed itself, it is going to consume completely. So this is all about what? Endosperm. So next we have is embryo. So embryo again, as you can see here, embryo development is again at the microphylar end, a microphyll end region, and of the embryo sac, where the zygote is actually situated. So what happens in embryo that you can see is, so the certain amount of the endosperm has been consumed as a nutrients during the development of the embryo. So this is one thing. True seed differ greatly, right? So there are certain early stages of the embryo development are similar in both monocot as well as in the dicot. But what happens? Despite the stages of the embryogenesis in the both, somewhat it gets varies. So the zygote will give rise to the zygote will give rise to the proembryo proembryo and subsequently to the globular heart shape. Can you see this thing? So it is somewhat globular. It is somewhat heart shaped, right? Globular. It is somewhat heart shaped and a matured embryo you will be seeing. So typically when I'm talking about the dicot here, so dicotyledons. So in dicot seeds or in the dicot embryo, you will be seeing the embryonic axis. So one thing what you are going to see, embryonal axis. So one thing is embryonal axis you are going to see, and two cotyledons is also what you will be observing. Okay. So another thing is you'll also see the epiblast region here. Then what else you have? That is hypocortical region will be seen, which is actually the terminal region. Okay. And you will also see subsequently, you will see what? Globular. Then subsequently, you'll see this radial part, root tip part, and coleorhiza part in the posterior way. So these root cap, it ensures it is going to cover this radical region. Okay. So which root cap? So root cap will actually help in penetration of the root. So this is all about the dicot. Whereas in monocot, what happens? So that is monocotyledon. So in monocotyledons, so here it is going to possess only one cotyledon. So here, how many we had? Two cotyledons. Here we only have one cotyledons. Okay. And they actually are grass family. So they are called as scutellum, right? And at the lower end region, what you are going to see is the embryonic axis will be having what? Again, the same thing. It will be having the radial cap region and radical region you will see. Then root cap will also be absorbed, which is going to enclose in an undifferentiated sheath, which is called as coolorhiza. Clear? So this is one thing. So as a result, you will also see, you will not going to see here the portion of the embryo here that we are talking, right? Huh. So you are actually going to see somewhat a leaf-like structure here, which is actually called as coulophyte. Okay. So this is what you are going to see in monocotyledons. Coming to the seed part, once they gets developed into seed, you just need to understand that non-albumin and albuni. See, so one is non albuminous and another one is, or it is also called as X albuminous, or this is what you need to understand. uh where it is, yeah. So as you have seen that in a simple way, so where exactly the storage of food reserve takes place, in the embryo, simply you will be seeing that there will be a reserved food and all, right? And seed coat you will also absorb. So matured seed may be either non-albuminous. Okay. So non-albuminous seed will be having no residual endosperm. It will be having what? No residual endosperm, as it completely consumes the as it is going to consume completely the endosperm during its development. Suppose here if you are seeing, okay, so therefore what happens, like wheat, maize, and barley all, so you will see this albumin In that way, what happens? This endosperm has been consumed completely during the embryonic development. And in some, you will actually see a kind of residue of this endosperm. So those are actually called as perisperm. Okay. So this residual, that is the peristent nucleus, is actually called as the perisperm. Then during unfavorable condition, some of the seeds, they actually get into this stage called as dormancy stage. So what is dormancy stage? The embryo may enter into a state of inactivity, which is actually called as dormancy state. And during favorable condition, they will germinate. Okay, this is one thing. Then the wall of the ovary, so that gets developed into the wall of the fruit. So basically what happens, the wall of the ovary will start develop into wall of the fruit processes stimulate simultaneously. So the wall of the ovary which develops into the wall of the fruit, right, is actually called as pericarp. Okay, which is actually called as what? Pericarp. Like if you have seen the apple, orange, mango, and all, so you will actually observe that what? There is a fleshy region, right? Which normally we eat. So that is actually called as the pericarp. So many fruits have evolved this mechanism for for dispersal of the seeds. So this is one thing. So another point is we have two types of fruits here. One is, sorry, false fruit and true fruit, right? So what happens is the thalamus, okay, the thalamus also contributes in the fruit formation. Such fruits are actually called as false fruits, where thalamus contribution is there. So this is the thalamus region. So if there is a thalamus contribution, okay, so like in few species such as apple, then strawberry, these where the thalamus also contributes in the formation of the fruit. So these seed fruits are called as false fruits. And most fruits, however, develops only from the ovary. They are called as what do you say that ovary. So they are actually called as true fruits. Okay. So although in most of the species, what happens is the fruits are result of the fertilization. So therefore, only few species, in which this fruit develops without fertilization. Such fruits are actually called as parthenocarpic fruits. So parthenocarpic fruits are nothing but the fruits which are developed without fertilization. Like banana is one such example for it. So this is about general aspects or basic aspects on fruits. In depth, again, I'm saying you in depth if you still want to know and all, the in-depth video is already there in the YouTube channel. So do go there, watch the video. You will get the in-depth knowledge of that thing also. So the last part is apopixis and sorry, apophysis and polyembryony. Right. So all those seeds and generally what you can see is they're produced through the fertilization and a few flowering plants such as some species, of grasses and all, they have evolved a special mechanism. So what is that mechanism? So to produce a seed without fertilization. So they are actually produce seed without fertilization. So they are called as apomix. apomixis, apomixis. Okay. So whereas here what happens, you will see this in the polymorph. So in the polyembryony, so you will see the occurrence of more than one embryo. Here you are going to see the occurrence of more than one embryo, where in a seed, okay, it is referred to as the polyembryony. So take off some examples such as orange and all. There you will be seeing more number of seeds, right? So they are called as polyembryony. Okay. So even the seed may differs, like from one seed to the shape, size, and all also might get differed. So yeah, this is all about the apomixis and polyembryony. So this is something called summary of the entire chapter, just in a one shot. And if you want to know even in detailed, I have also made an in detailed lecture in this chapter. So go watch that, and yeah.